Animals differ in what they eat. Because of that, they have to adjust, not only behaviorally, but physiologically as well. The physiology of the digestive system of animals varies. They vary in terms of the morphology and functions of the gastrointestinal tract, and their variation seems to depend on the nature and chemistry of what they eat. From this illustration, it can be seen that the human digestive tract is relatively small. Compared with that in the pig, an omnivore that is often regarded as a model for humans, the human large intestine is much reduced. The dog intestine is capacious but relatively short. The human large intestine is also small compared with the orangutan. The kangaroo, a non-ruminant foregut fermenter, has a large sacculated stomach, whereas the hindgut fermenter, the horse, has a capacious, multi-compartment large bowel. The koala, which consumes only leaves that are rich in tannins and volatile oils, has an extensive large bowel and reduced small intestine.
As you may have noticed in that illustration, animals have distinct anatomical differences in their digestive tract, and it seems that it is related to the chemical nature of what they eat.
Digestion in herbivores. Many domestic animals are herbivores, that is why it is very important for us to understand their digestive physiology. Herbivores have the following distinct characteristics. They feed on plants. Their mouth structures are adapted to gathering, cutting, and grinding plants and plant parts. They rely heavily on microbes for digesting or fermenting cellulose, and they mostly absorb volatile fatty acids, or VPHYs, instead of glucose. It is very important to understand what fermentation means, as most domestic animals, especially the herbivores we have been talking about, largely depends on it. Fermentation is the enzymatic decomposition and utilization of foodstuffs, particularly carbohydrates, by microbes. Fermentation takes place throughout the gastrointestinal tract of all animals, but the intensity of fermentation depends on microbe numbers, which are generally highest in the large intestines. Thus, the large intestine is quantitatively the most important site for fermentation, except for ruminants, in which the forest stomach has the highest microbial concentration. Furthermore, there are major differences in the contribution of fermentation to energy production of different species. In carnivores like dogs and cats, and even in omnivores like humans, fermentation generates rather few calories, but in herbivores, fermentation is a way of life. Cellulose is a common constituent in the diet of many animals, including humans. However, no mammalian cell is known to produce cellulase. Several species of microbes in the gastrointestinal tract produce celluloses and digest cellulose. The major end products of microbial digestion of cellulose and other carbohydrates are volatile fatty acids, methane, hydrogen, and carbon dioxide. These microbes also synthesize vitamin K and certain B vitamins, and they themselves serve as nutrient source, especially protein, for ruminants. Among the professional fermenters, two distinct strategies have evolved: the cranial fermenters or ruminants, and the caudal fermenters or cecal digesters. The main difference between the two groups is the position of their fermentation vat relative to the true stomach and small intestine. Cranial fermenters or ruminants have a large, multi-compartmented section of the digestive tract between the esophagus and true stomach. These forest stomachs house a very complex ecosystem that supports fermentation. Some examples of ruminants are cattle, sheep, deer, elk, carabaos, and buffaloes. Caudal fermenters, also known as cecal digesters, are similar to dogs and humans through the stomach and small intestine, but their large intestine, where fermentation occurs, is complex and exceptionally large. Examples of cecal digesters include horses and rabbits. If we compare cranial and caudal fermenters, we will see that both cranial and caudal fermenters can efficiently digest energy from cellulose. However, only caudal fermenters are able to utilize sugars directly from the food they eat. On the other hand, only cranial fermenters have the ability to use protein from fermentative microbes. To continue with the comparison, let's look at this illustration also shown in our previous video. We can see here that the cow's digestive tract is larger in terms of total volume. However, the largest compartment for cows is the rumen, while for horses, it is the cecum. This highlights the fact that their fermentation area differs.
If you want a more detailed discussion on ruminant digestive physiology, you may view the video posted here in our FB group, or you may watch this link. Okay, so thank you very much for listening to this short video. I hope you have a great time.